All right, so now we're going to move on to our second part of Lecture 2, which is sort of a sidestep. And I just wanted to focus a little bit on cloning because cloning is becoming really part of genetics. So with cloning, you are going to basically use vectors. Vectors are sort of, you can think of them as genomic circular plasmids, and these are going to be used in cloning. Now, we talked last lecture about EcoR1. EcoR1, remember, is E. coli restrictive enzyme 1. And E. coli, EcoR1 is going to be able to cleave DNA into fragments. And so what this does, it creates what are called sticky ends. And so our sticky ends are here, and they kind of can overlap and stick to each other. And so what that will allow is your EcoR1 will cleave your DNA into pieces, and then those ends will be able to come back and religate in a process called annealing, and they will stick together. So you can use these techniques in order to create the right sticky ends so that you can bring together genes that you want to incorporate. So you can bring in a gene from that you put in a bacterium, for example, in a bacteria plasmid, and you can recombine them to bind to human DNA. And this is going to be called recombinant DNA. So human DNA can be cloned, and these genes can be put into a bacteria and to make a library. You might have heard the term library. It can be a yeast library as well. Yeast cells do the same thing. They basically are just going to replicate whatever you stick in them. So you can stick in a cystic fibrosis gene, for example, and you're going to make more of the cystic fibrosis gene through replication. Now, there are different cloning vectors. Talked about plasmids. There are also bacteriophages. Bacteriophages are going to be viruses that infect bacteria. These can bring genes, so they can transmit genes between bacteria. We have cosmids, which are phase plasmids, and they are about 50 kilobases long. We have yeast artificial chromosomes that are much larger, and we also have bacteria artificial chromosomes, or BACs. And each of these are just tools that somebody would use to clone genes, to replicate genes, etc. So what do we use clones for? We can create thousands of copies of a gene for analysis. And so this is going to be important for trying to identify what is the gene, what is it doing, what's its function. You can also produce engineered therapeutic products. So insulin is made through cloning, different interferons, interferon alpha, interferon gamma, human growth hormone to treat genetic diseases. Clotting factors can be made to treat different deficiencies of clotting factors or coagulation disorders. Tissue plasminogen activators is going to help to dissolve blood clots and help to prevent heart attacks and strokes. So all of these are made through cloning. So just as a southern blot, so southern blot, or which is also referred to as an audio radiogram, is going to be used, for example, in pedigree analysis, used to detect insertions and deletions of DNA, because again, if you have a change in the fragment length, you can see that change based on the size of the fragment on the actual gel. So if you have different mutations, you can pick that up using southern blotting as well. This is all done by analyzing restriction length polymorphisms, or RFLPs. Restriction length polymorphisms is just what we had just talked about. It's that E. coli. It's E. coli R1 making different lengths. So you have the Eco R1 that will clip off different sizes based on a consensus sequence, that it recognizes. So EcoR1, and there are hundreds of restriction enzymes, so you can make various sizes of your DNA, chop them up into various sizes, run them on a gel, and then you'll have your person 1 that will have certain bands, person 2 will have certain bands, etc., using the same enzymes. So that's all RFLPs are. So RFLPs are going to be used to identify cystic fibrosis, Huntington's disease, type 1 neurofibromatosis, for example. So this is just an example of a pedigree. So we have 3 generations of our pedigree shown here. And this is just showing restriction enzymes that make these 2 sizes of DNA. So we have a 5 kilobase size band of DNA, and then we have a 2 kilobase band of DNA. And so on chromosome A, you can see that you have your restriction sites, which are here and here, and so you get a 5 kilobase band. And then on chromosome B, you have more restriction sites, so you get a 2 kilobase band and a 3 kilobase band. Now, what you're looking for here is inheritance of a mutation. So you can see that you have your first generation, maternal and paternal. The paternal is the square, maternal is the circle. And so both of these have some children, and one of those children have the disease, so they're affected. And so when you look at this through restriction fragmentation, you can see that both parents, so this band reflects the parents, both parents have 2 bands, right? They have a 5 kilobase and a 2 kilobase band. And so they are heterozygote. And then you can see that you have the offspring that are not affected are going to have either 1 or 2 bands. So this one would be a homozygote for that chromosome A, and these would be heterozygote for A and B, similar to the parents. The affected was inherited a chromosome B from both parents, and so this person became affected. Now you can see that this 4 sibling got married, and he had a heterozygote phenotype. He married somebody who was homozygote for the unmutated chromosome A. And so now with their offspring, they have 50% are carrier of that, and 1 is unaffected. And so you can see through this, by using southern blotting, you can easily track the mutations in this pedigree. All right, so for other uses for cloning are to identify tandem repeat polymorphisms. You can use satellite DNA to measure diversity. The variation is in the number of repeats within a different region, and this has substantial variation from person to person. Your mini satellite polymorphisms are going to have variable number tandem repeats. Microsatellite polymorphisms are going to have short tandem repeats. And these polymorphisms are useful in paternity testing and for identifying criminal suspects. So this is an example of a forensic sample. So again, you're using different repeats to identify potential potential criminal for a particular crime. And so, for example, these are DNA ladders. And these are just known, DNA ladders are simply known sized fragments of DNA. So they're sort of a standard. And so you can see here that you have across 1, 2, and 3 bands that are associating with a particular size, and you can determine that size based on the DNA ladder because you know the size of the band here, for example, and here, so you would know what the size of that band is that runs about the same length on your gel. And then you have, of course, some differences here. So in this one person, you have a band here. You don't have that in your 2 and 3 sample, but in your 2 and 3 sample, you have 2 bands here, which you don't have in your 1 sample. So depending on who is the sample that was taken at the scene of the crime, for example, that was sample 3. So this is the crime scene. sample, you would then say person 1 is not associated with the crime or cannot be associated with the crime because the repeats don't match the crime scene, whereas person 2, the repeats do match the crime scene, so they are potentially the person who committed that crime. Okay, so these restriction fragment polymorphisms are going to have sequences of DNA which are going to be different at different restriction sites. And so this is just showing different restriction sites. These little triangles here are just showing you where the enzyme, whatever enzyme you decided, EcoR1, whatever, is going to cut. And so you're going to have different lengths that will be run on the gel. So here you would have a cut here, and then you would have another length here, whereas on this bottom one, you would have a much longer fragment because they don't have a sequence in between, and so your EcoR1 can only cut where it can identify its consensus sequence. We have tandem repeats, which are going to be short fragments of DNA that are repeated in tandem. So this is CAG over and over and over again. You have microsatellite and mini satellite repeats of these. And so these can be used, of course, and they're going to be different from person to person. You also have copy number variants. These are going to be larger repeated segments. So these are going to have 1,000 base pairs. And so you have, again, differences in these larger repeats also from person to person. Single nucleotide polymorphisms, or SNPs, are single base pair variations in the population or in the genome. So this difference at this location of the genome is single base pair, and so this is referred to as a SNP. Now, methods to detect variation, we showed southern blotting, but southern blotting is sort of an old way of looking at DNA. Nowadays, you use PCR, and one of the benefits of PCR is you don't need a lot of DNA to start with, and it's very rapid. So basically what PCR is doing is you take a very small amount of DNA and it's going to replicate it, so now you have a large amount of that same DNA. And then you can do different restriction enzymes similar to what you did in your southern blotting, or you can make different probes and you can identify different sequences within the DNA. This is going to have primers. Primers are going to bind to DNA and going to replicate that DNA or amplify it. Some of the disadvantages of your PCR is you have to know what sequence you're trying to amplify. There's also a high propensity for contamination of nucleic acids from the environment, and it can't be used for large deletions. So another method to detect variations at the DNA level is called the Sanger method. So in this case, you have what are called chain-terminating dideoxynucleotides. And so you can think that when you're trying to sequence your DNA, if you have, for example, it's kind of showing this, maybe I can blow it up a little bit. You have your ATP, CTP, GTP, and TTP. You're adding your bases to the actual reaction. And then wherever, if you add, for example, the DDATP, wherever there's an A, it's going to stop. And so you know you have 5 bases and then you have an A. So you can say, for example, I have 1, 2, 3, 4, 5. You know this 5th position is an A. You don't know what these are yet. And then you would add your T, for example, and say you got a T here. And so now you know you have a T, question mark, question mark, question mark, A. Then you add your C's. So now you know you have a T, and then you have a C, and then you have an A. These are still question marks. Then you add your G, and you have a T and a G, question mark, C, A. Add another G, G, C, A. So this is a way to identify what the sequence is of your genome. It's a very slow method, but you can actually do it. The problem is, as I said before, it's slow, error prone, and we do have newer methods now to do that. We have what's called high throughput DNA sequencing, which is more rapid than the traditional. This is going to be done on a glass slide. So whereas in the Sanger method, you would do it one at a time, the glass slide allows you to amplify your DNA fragment that you want to sequence, put it on the glass slide, and then add all the bases at one time, and they can all be labeled. And the label, the fluorescent label, would be used. So you would have your A would be pink, G would be blue, for example, and then you would be able to read this, and then you can take these DNA sequences and plug back in their actual sequences based on the fluorescence of the dye at the particular locations. Another method for measuring DNA variation is microarrays. And so microarrays are referred to as chips. We now have protein chips, RNA chips, DNA chips, a lot of different types of chips. And so basically you're going to put on one of these chips, which are very small, smaller than a slide. You can put your known oligonucleotides. For example, if you know you're looking for sickle cell anemia or cystic fibrosis gene, then you put them on a slide. And then you're going to label your single-stranded DNA from a patient. You're going to separate the DNA. You're going to label them with a probe, add them to the slide, and then if they bind or anneal to a particular area, then you can say this patient has this gene or this mutation, for example. You can add several million SNPs on a single microarray. It's a very powerful tool to be able to see multiple things at the same time. This is an example of a DNA microarray chip. This is a normal fibroblast DNA in red, Niemann-Pick disease fibroblast DNA in green, and the yellow is an overlap. So if you go through this, you can see that the normal DNA, you have these little areas of these brighter areas of red, and then you also have these areas of green, and those would be specific for the Niemann-Pick disease. And so you can go back to that spot on the microarray and say, okay, what sequence is glowing green and what is the function of whatever that protein is at that particular location on the microarray. So it's a very powerful method to identify proteins or sequences that are unique to a specific disease. So all these greens would be unique to Niemann-Pick disease. So those would be what you would try to focus in on as causing the symptoms of the particular disease.